Thank you, Cheryl. I've got one housekeeping piece to confer before I ask Amy to present her work. We found a key card from the Monaco Hotel. So if you're missing yours, you might want to see Patrick in the back. And now I'll ask Amy LeBlanc to talk to us about the Comparative Oncology Trials Consortium. Thank you.
Okay, so I have the distinct pleasure of being the last person to talk today. So I know that there's probably some attractive food and drink options waiting for us. So I will try to make this brief and hope to tell you all about the pretty amazing opportunity that the NCI's Comparative Oncology Trials Consortium represents for our community and for this effort. And so my hope today is to give you a sense of our structure, our mission, and our goals, and our view for the future. So the discussion points are simply taken out of the IOM agenda planning documents, and that will focus today on how we operate and whether or not we believe that the current structure meets the needs of the drug development community and what challenges we face moving forward. And I'll have a couple of new and innovative strategies that we could potentially employ to improve the relevance and accessibility to those involved in drug development while also fulfilling our mission at the NCI. So first, how do we operate? So we are a component of the NCI Comparative Oncology Program. Certainly our program is engaged in this variety of efforts. Our webpage has been recently updated, so you can take a look at that. We also are engaged in some basic and translational imaging research and bench drug development activities, as well as my program manager, Christina Masco, is the executive director and sort of manages the day-to-day operations of the CCOGC, which is the biospecimen repository you heard about earlier. So we have a variety of activities, and they're sort of broken out here. One of the things to also just keep you aware of, sort of continuing along the imaging theme, and that our collaboration with Pete Chwicky's group is growing and a really exciting opportunity, especially since our clinical trial network, 6 of our sites have really amazing molecular imaging facilities in-house. And so that's something that I hope to see grow in the future, as our clinical trials certainly have a history of being in small molecules and you know, having a PK/PD heavy design, but I would love to see us more engaged fully in the opportunities to investigate cancer imaging through our network's resources. We are also starting a new comparative brain tumor initiative this fall, and so that's still sort of under development, but I think you've already gotten a sense today that drug development with respect to brain tumors is something that is growing, and we recognize the amazing expertise amongst members of our clinical trial network, and that being able to leverage that along with all of the imaging expertise, I think, is a really exciting opportunity. So just for folks who might not know necessarily where we sit in the big machine of NIH, so obviously there's the 27 distinct institutes and centers, of which NCI is one, and then you have in this diagram things broken out with respect to what's extramural research support and what is intramural. And then within the Office of the Director of the CCR, our program sits directly underneath that office. Right there. So why would people consider the COTC mechanism and infrastructure an attractive option for drug discovery and drug development? So I think first and foremost, it's not lost on me that I get to stand up here and talk with you today because of how amazing our investigators and our sites are. So there's really no ability to describe the depth of expertise in clinical trial design and the amazing patient care and dedication to our studies that goes on there. We have criteria for membership, and that helps us really maintain a high standard of excellence. They have access to patients. They have the ability to reach out through clinical trials offices and social media and a lot of outlets to really make sure that the community of the pet owning public knows what their options are. We offer clinical trial support that is dedicated. We're not a CRO. We are politically neutral and really want our membership to really feel like this is their consortium and that we are here to really support the mission. And luckily, because we are an NCI-supported intramural endeavor, we have the ability to, again, leverage the existing NCI resources. We have a purpose-built C3D data management, a web-based reporting system, and we have access to amazing opportunities for collaborations within the NCI, our own lab, but other labs within our institute. And so having that visibility, I think, is a really valuable attribute of our program. So this is, again, one of maybe the big win of my first year here within the NCI is that this map has recently been labeled correctly, and that a handful of our states were wrong for a long time. And so I have to have a shout out to Carolyn Henry, who's probably like telling me to stop bringing this up, but she thankfully pointed out that the map needed to be correctly labeled, but eventually we'll get a landmass for Guelph to sit under as our Canadian site. That we'll put that for next year. That'll be one of the goals. Next year is to have Canada be fully represented there. So I put this together actually when I first joined the program because I really needed to understand sort of the amazement of paperwork and agreements and things that help keep us legal. So basically, when our program initiates an agreement to do work with a sponsor, we have a confidentiality agreement, a protocol, and our sites tell us what they need to be paid to do these studies, and so we pass along that budgetary information to the sponsor so they can sort of review the necessity of the protocol meeting their needs and the questions that, again, we're striving to answer to help advance their particular agent. And then occasionally we also act as drug distributor. So there's a materials transfer agreement that goes between the sponsor and our program so that we can make trial packages and get paperwork and labeled tubes and study drug from our place to our sites when they enroll a case. And then between us and our sites, we have a memorandum of understanding that also includes confidentiality agreement information, and the agreements between us and our sponsor and the agreements between us and our sites mirrored the same language. So everybody's on the same page about how data is collected, who owns the protocol, and who owns the data that's generated, and how IP that might be generated or might not be generated is handled. So C3D is our online data reporting package. So we, and I say we, I mean Christina manages C3D and has the ability to generate reports from that. And so we communicate that data that's collected in real time from the sites back to the sponsor. And then the sponsors pay the COTC sites directly for clinical case management. So they have to initiate individual contracts with each site that's participating. And then any correlative assays that are performed within the pharmacodynamic core, which is a really amazing set of expertise to allow dog-specific assays to be carried out in support of the work, that's also a separate contract. And then COTC sites maintain their own institutional animal care and use documentation. So again, study budgeting, it's a standardized fee structure that the sites have decided upon amongst themselves. It carries with it some investigator and technical support line items. There's separate budgeting for correlative assays. And then generally the study sponsor is responsible for providing the agent. And so this goes back to our comments earlier about how it's a significant amount of drug that's often needed for these studies in these size patients. And then often the study sponsor is the best person to be performing any pharmacokinetic metabolite analysis or any other biomarker discovery work. Protocols and consent, and this is just in here to sort of give you an idea of how very specifically we manage our trials and how we have this commitment to having everything lined out up front. So we have a commitment to excellence in the data that we generate with our sites. The protocol and the consent documents go through a lot of review by our membership with input from everyone up front. The informed consent document is tailored to the study agent and the procedures that are carried out within the trial. There's going to be discussion about protocol consenting and communications with pet owners tomorrow, but I will say that our consent document, not unlike anyone else's, includes adverse event languages of what is to be expected. It includes a summary of risks and benefits to the patients. Also important that our study sites don't go off protocol. which would represent a violation of the MOU. And so that's, again, just sort of a thing for the sponsors to understand, and that our sites are following a protocol, and that the data that is generated is not considered novel IP because the study sponsor owns the data. And then, as I mentioned, the sites maintain all of the NCI-COP approval documentation, and they're responsible for filing amendments to the protocol if things develop during the trial that necessitate such an amendment. Data management is done through an online C3D system that is specifically built for each trial. I'll just show you what that looks like. It's an Oracle-based system, so each case is monitored and data is entered in real time. Physical exam findings, correlative, hematologic biochemistry data, things like that are directly inputted in here. Follow-up procedures, concurrent medications, and the like. There's even pages in here that are filled out with FedEx tracking numbers, so we know what shipments are set on what day, so all of that can be tracked. We also convene a data safety monitoring board for each trial. It consists of a chair and then 4 members that are usually from non-participating institutions to eliminate bias. We have quarterly discussion of all the adverse events within the site investigators and keep the sponsors, of course, apprised of all that, and then have ad hoc discussions of things that come up unexpectedly. Okay, so do we meet the needs of the drug development community? I think just sort of using this very famous sort of diagram from the Hanahan and Weinberg iteration of the hallmarks of cancer, several of our studies that have been brought up today really meet the needs of these specific aspects of what cancer cells are able to do. And so we have several, documented endeavors where we've addressed these things. And so I think on this level, we really think long and hard about how we can design studies and work with companies that have developed or are developing drugs that address these individual specific aspects of cancer. One point about COTC trials in that generally we aim to look at getting data that is in response to a specific need, and a human is in parentheses here. I mean, I think that it's the line between human and veterinary drug development becomes sharper as you get into looking at licensing and labeling and registration trials. But early on in a molecule's development, I think if we really think long and hard about cancer, and we've talked a lot today about the similarities and things that we know and things that we don't know, but we can learn a whole lot about tumor biology and how a drug behaves in a dog, and that can be relatable to veterinary drug development or human drug development. So I would say that we really aim to have studies that are very rich with respect to asking questions that can really uniquely be asked in dogs and dogs only. Most models are helpful, right? Not one will solve them all. So we look, again, just as we've mentioned many times today, about focusing on tumor biology and drug target rather than just looking at histology. As Cheryl very nicely pointed out, there are lots of opportunities to refine dose, schedule, look at biomarker validation, as Doug pointed out earlier today. Many people have brought up the concept of combination therapies being where we need to be heading. And efficacy, although that might be the ultimate biomarker, is often not the primary endpoint of the studies that we do. But of course, it's always something that we like to see and hope to see. I'm going to walk you through an example of a study that is where it's been referred to many times today as and you'll hear about it tomorrow too, as a pick the winner design, where 3 candidates were sort of looked at out of a preclinical package, and the idea was to try to distinguish them in a dog model and have that then flow naturally into a human series of phase 1 trial to have sort of a nice validated 3 species package of data. So 2 of these agents were escalated. 1 reached an MTD, the other did not. And then 1 is certainly still going. An MTD was identified, and now we have a cohort expansion open. And so here are just the progress of how the cases were enrolled over time. And you'll see that because we have multiple sites involved at any one time, and these were agents that needed very careful assessment of toxicity, that the enrollment was actually quite slow because we needed to be very careful about how the toxicity was assessed. And these are PD heavy cases. And so we're asking a lot of our sites to collect multiple tumor biopsies and PK curves. And these cases take a lot of work. And so it is a testament to our sites that they were able to help us out with taking these 3 agents in real time and randomly filling the cohorts as we move forward. This is a waterfall plot that shows you the 3 agents in various shades of green, blue, and purple. And I think the thing to notice here is that, when we're looking at these 3 agents, trying to identify how to distinguish one from another, we wouldn't just do it based on response. We would look at other criteria to help discern how to tell them apart. So this is where I get into one of these things is not like the other. If anyone is old enough to remember this, we have to have a veterinary correlate for it too. So, I couldn't find a good dog one, but I like this one with the owls a lot. So in this case, what I'm about to show you is not so much that we were looking for a winner. What we wound up finding was an oddball. So we have these 3 agents again, the MTD not being reached for the compound that we refer to by the last 3 digits as 400. But the response rates defined as PR or better based on the VCOG lymphoma scale are listed there. And then contrasting that to the response at the MTD, you get the sense by just looking at these numbers that there might be something different about 744. And then when we look at trying to break them out with respect to asking whether there's a dose-response relationship, well, it certainly appears that there is within 776 and 400, but there's not necessarily within 744. And these dogs, again, this was an open enrollment, so we had previously treated and naive patients coming on this trial. I don't think we had the statistical power to break them out into individual groups, but we're not done with this study yet, so this is considered a preliminary assessment of the data. But right away you say, okay, one of those things doesn't look like the other 2. And then when you look at differential, now this is tumor PK, tumor drug concentration at day 6. These particular agents are given daily for 5 consecutive days as an IV infusion. And then an entire lymph node, these are all lymphoma patients, lymph nodes taken out at day 6. And then we're looking at drug concentration with respect to the best response plotted as a % decrease in disease on the y-axis. And so you can see that 744's scale is 10 times higher than what you'll see in tumor drug accumulation for the other 2 agents. And then when we look at the PD readout for these drugs are thought to has their main mechanism of action be a topoisomerase 1 or TOP1 inhibition. So gamma H2AX would be the logical PD readout for these agents. So again, 744 isn't behaving, at least from a PD readout, like the other 2. So I show you that example because that's not what we expected to see. And it was exactly the reason to do a study like this, in that the agents did not look all that different coming out of preclinical mouse models. There was admittedly a hint that 744 may not have the direct relationship with the gamma H2AX signal post-therapy, but now we are in a position to offer a critical piece of information and really now have the opportunity to take this dog data and then go back and mine it for the relevant biomarker for 744. And in fact, the IND for 744 is being held until we can actually finish the trial and find the appropriate biomarker before initiating human trials. So how do we serve the community? We provide centralized trial management at no cost. Because we are an NCI-supported intramural effort. We have PD-rich studies that occur in naturally occurring cancer, and we can study both patients with naive and resistant disease because of the direct access to amazing expertise, reagents, and assays to support these trials. We do have a link to comparative cancer imaging based not only on our efforts in Bethesda on the campus to bring dogs with cancer to the NIH campus for imaging, but through the expertise in imaging amongst our sites. And we have a basic science laboratory component to our program that allows us to leverage, again, very unique resources geared towards metastasis biology. So what are our challenges? Well, you saw that previous slide about numbers of agreements and the time needed to execute them. So, I think we can handle most agreement terms relating to IP because of the terms of our MOU. But it certainly is a long time to ask contract after contract after contract to be executed between us, study sponsors, and sites. There are competition for cases. And so you've just seen several examples of comparative oncology studies that are occurring at our network sites. And it's hard, when individual sites have been so well enthused about their own work, how do they balance? It's a challenge. How do they balance putting a dog on their study versus one of ours? And this slide you've seen a bunch of times today. And I think, again, sort of along we might be a victim of our own success, that people are starting to use it, or various likenesses of it, for their own work. So it's nice to see these things come out in the literature, because it means that we're making an impact. But this is sort of messy. And so all these agreements, we try to figure out ways that we can try to streamline this to make it easier for pharmaceutical companies to work with us. The trial development process of I'm starting to beat a dead horse here. So there's a lot that goes into these things, and I think that that's a testament to the quality of the data that we turn out. Again, it's just a testament to the expertise of the folks that we work with. And I think now, we have to think about what strategies could we employ to improve accessibility and relevance to our mission and that of the drug development community and the cancer community. We spend a lot of time asking questions. Old questions were simple. Will it make dogs sick? Do dogs get the same kinds of cancer? Does tumor histology predict response? We've heard a lot today, and I'll just have the ability to wrap it up to say, our questions are getting more and more sophisticated as the need to really refine drug development and limit the attrition of drugs as they progress through the paradigm. The strengths of a multi-site consortium, some of them are obvious in that, if you need large numbers of patients with a specific disease that may not be all that common, and that's directly translatable to humans based on a number of factors we know are in place with respect to biology and genomics, that's obviously a strength. Minimizing bias, either geographical or linked to an investigator. One thing that I really hope to see happen more is that we can support early stage investigators who can leverage our administrative support to conduct a large trial that they otherwise would not be able to do that. And then to vertically integrate our in-house preclinical drug discovery tools and methods into the development path of a novel agent. I think we have the ability to do that. New things that we think about, emphasis on novel study design. So combination therapies is something that we're going to spend a lot of time thinking about and implementing in the coming months to years. Thinking about this concept of safety data generated in pet dogs and how to manage that perception and risk of perception as agents are examined by the FDA. Thinking about contract core to facilitate the agreements and trying to streamline that process. And then spending time looking at directed programs in addition to clinical trials, because our network does a lot of clinical trial work and they do it really well. And so maybe it's time for us to think about more directed program efforts rather than just continuing to bang away at multi-site clinical trials as our main effort. So as I've mentioned, we're thinking about an exceptional responder program. We're definitely moving forward with a brain tumor initiative and with a comparative cancer imaging fellowship. So, I hope that I've gotten across the message that I believe in our program and our network as a high quality integrated multi-center network that conducts studies that are of very high quality in response to a specific need. And comparative oncology efforts have certainly flourished thanks to Dr. Khanna and all of the folks that work so hard every day to advance the message of what pet dogs could potentially offer. And we need new initiatives that extend beyond clinical trials. And I think we'd all agree that ongoing molecular validation of canine cancer and those efforts will, certainly deeply contribute to our knowledge moving forward. So with that, I want to thank everyone who's here and also who has helped me succeed over the years. And I look forward to working with all of you more in the future. And I'd be happy to take any questions. Okay. Thank you.